Thank you so much. It's really a great pleasure to be able to present some of our work to you today. What I was asked to do is tell you a little bit about my career, then discuss why it's important to strengthen the bridge between life sciences and technological innovation. And to do that, what I thought I'd do is give a number of examples of what we're doing, which does, I think, bridge that gap. And then finally, I was supposed to go over what Scandinavia can potentially learn from the U.S. ecosystem. And again, we probably can learn a lot from the Scandinavian system too, but I thought again, the examples that I'm going to talk, which are about what we've done on technology, I think will give aspects of that. And then I'll just sort of summarize, some of those points. So to start a little bit about me, I'm a chemical engineer, and I got done with my degrees in 1974. And when I finished, pretty much all my colleagues went to the oil industry because that's where all the high-paying jobs were in 1974 in the United States. But I wasn't very excited about that, and I ended up deciding to work actually in a hospital. And I was the only engineer in a hospital. And the reason I did it was I was interested in seeing if there was something that I could do with my chemical engineering background that would help people, help their education, help their health. So I went to work for a man named Judah Folkman. He was a surgeon. Sometimes I always tell the story that, I tried to get a lot of jobs in hospitals and nobody would hire me. And then one of my friends told me I should write to him because sometimes he hired unusual people. And he was nice enough to hire me. And the idea that he had that I started working on is shown on the next slide. And his idea was that if you could stop blood vessels from growing, maybe that would be a whole new way of stopping cancer. So what you see in this graph and this picture is that a tumor, it starts out being very, very small. And then it gets bigger and bigger, but it can't get beyond about 1 million cells or a millimeter cubed. And the reason that happens is because it runs into a nutrition problem. The tumor can't get oxygen or get rid of waste. But what Dr. Folkman said is the tumor secretes a substance called TAF, tumor angiogenesis factor, and that causes the surrounding blood vessels to grow. And they grow to the tumor and it feeds the tumor. And that solves the nutrition problem. So the tumor gets bigger and bigger and bigger. And ultimately, it not only gets big, but it can spread through those blood vessels. That's a process called metastasis, and that's what kills people. So when I started working for him, nobody believed that this was correct. And my job was to prove it was correct. And in so doing, isolate the first substances that could stop this from happening, which we call angiogenesis, anti-angiogenesis. So there are two problems. One is that you have to find something, but the other is that you have to have a bioassay. And there was no bioassay to even study blood vessels from growing. So on the next slide, we tried to invent one. And what we tried to do, and this all is like super interdisciplinary work between the life sciences and with blood vessels and between technology, like I'm going to talk about in engineering, like polymers. But the idea we had was we'd put a tumor in the eye that would cause blood vessels to grow. But the problem is it took about 9 weeks for that to happen. So we wanted to have what we call a slow release polymer that could release any of the molecules that I was isolating, and all of them were big molecules like proteins and peptides and things like that. So we wanted something that could release this and not cause any harm to the eye, and that would release it for over a 9-week period. And so again, I wasn't any expert on polymers, nor was he. So, we asked different people who were what they thought, and they all told us it was impossible, that you couldn't release large molecules from polymers because that was like walking through a brick wall. The literature actually said the same thing. It said, if we go to the next slide, that the use of polymer matrices for slow release systems has been virtually restricted to small molecules. And sometimes what I tell people, which is true, is maybe the only reason I was able to solve this problem is I hadn't read the literature. So what I did is I spent several years working in a laboratory experimenting kind of Edisonian-like, again, technology-wise, with different approaches to make a slow-release system. And I found over 200 that didn't work. But eventually, next slide, I discovered a way to make little microspheres, and now, as I'll show you later, we can do nanospheres. Here's a picture of one, and here's one cut in half. And on the next slide, we published in Nature in 1976 the idea that you could do this to release really any molecule for the first time for over 100 days. Now, when we did this, it received enormous scientific skepticism. I remember giving a lecture on this to a group of very famous chemists and chemical engineers in 1976. In fact, it was the first lecture I ever gave, at a public meeting, and I was very nervous. So when I got done with the lecture, I thought I did okay because I didn't stammer too much and I didn't forget too much what I was going to say. So I thought all the older scientists, being nice people, would want to encourage me, this young guy. But when I got done, I stepped off the podium and a whole bunch of them came up to me and they said, "We don't believe anything you just said." They said, if we go to the next slide, that large molecules can't slowly diffuse through solid polymers, which was what people had said earlier. And they also said the solvents we're using, they'll destroy whatever we put in. And actually things went downhill from there, if that's possible. I tried to get grants, and my first 9 grants were rejected. I really liked actually being a postdoc, but a lot of people, my friends, said it's probably not good to do that forever. So I started to apply to faculty positions. And like I said, I'm a chemical engineer, so I applied to chemical engineering departments. And no chemical engineering department would hire me. They all said, this biology stuff you're doing doesn't make any sense. So I ended up getting a job in the nutrition department. The reason I think I got it was the man who hired me, Nevin Scrimshaw. He was kind of like a benevolent dictator kind of department head. He liked me, so he hired me, but he didn't bother to ask anybody else in the department what they thought. That still might have been okay, except for the fact that that the year after I joined the department, he left. So a lot of the senior faculty told me, they gave me advice. They said I should leave too. I'll illustrate that on the next slide. This was actually from Mike Marletta, who's a very famous chemist. He was an assistant professor with me in the department. He later became chair of chemistry at Berkeley and is a member of the National Academy of Sciences. And he's just saying here one time he went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for polymer drug delivery, he blew a cloud of smoke in my face and said, I better start looking for another job. And Mike said he was in a Fellini movie. So that wasn't so good, but I kept persisting. That's important, I guess. And just going back to that assay that I showed you, if we go to the next slide, this is what we wanted to do. And on the next slide, this is a little bit of a hard slide to see, but we published in Science that we actually isolated the very first inhibitors of blood vessel growth. And so what you can see, if you put a tumor, this was 9 weeks after the start of the experiment, in the rabbit eye, normally, if you don't have this inhibitor, we called it CDI, a sheet of blood vessels grows over the polymer. Here you see the tip of it to the tumor. In fact, if you looked at this 3 weeks later, it would be huge. It would be out of the orbit of the eye and it would kill. We sacrificed the animals before that. But when we put the inhibitor in, and by the way, we did 2,000 eyes, we found hundreds of things that didn't work. But we did find one fraction that did work. And it showed for the first time that blood vessels could be stopped. You can see here, they're sparser, they're lower, they avoid the tumor. And about 60% of the time, these tumors never even grew at all. So that was very exciting and showed that you could stop blood vessel growth, stop tumor growth. Still, it took 28 years and many companies working on this before the first blood vessel inhibitor would be approved by FDA. And this is shown on the next slide, that you see these approvals. Avastin, which was the first one 28 years later, this was great work done by Genentech. spending over $1 billion, I'm sure, to do this. Avastin's been one of the best-selling drugs in history. And this keeps going. Many drugs now have been approved for, and many are blockbusters, not only for treating all kinds of cancer, but treating diabetic retinopathy, which is a terrible eye disease, macular degeneration, which is also a very bad eye disease. In fact, there were no approvals for this before these eye diseases for any drugs before this. It had only been treated by lasers and other things, and they didn't work very well. I also thought that that not only might this work we did with the polymers be useful for treating isolating these angiogenesis inhibitors, but they may be useful in their own right for treating different diseases where the half-lives are short and where the molecules are large or ionic. So one of the things that's key, and this is a very important thing when people ask about creating technology, creating companies, is we wanted to get a, Dr. Folkman and I thought we'd try to get a patent. And in the 1970s when we did this, nobody in Boston Children's Hospital, which is a very good hospital, ever had a patent before. So we filed for the first one. And 5 years in a row, it was rejected. The patent examiner rejected it. He said he didn't really understand the science, but it didn't matter because he was calling the shots. So everybody told me I should give up. But as you can see, I don't like to give up easily. But science wasn't working. So I tried to think, how could I convince the patent examiner to allow this patent, legally, of course. And I, as I said in the beginning, when I first started doing this, everybody said it was impossible. It couldn't work. So I wondered if anybody ever wrote that down. So I did what's called a science citation search, looking back at our 1976 paper, science paper, a Nature paper rather, to see what people said about it. And I found some very interesting things. We'll go to the next slide. And here, I had no idea this was written. So the Nature paper was written in 76, as I mentioned. I was doing this now in 1982, but I found 5 of the top polymer chemists in the world had written a review on this field. It was a very, very young field at the time that we helped start. So they said, generally, the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example, proteins, could be released by such a technique because of their extremely small permeation rates through polymers. However, myself and Dr. Folkman have reported some surprising results, and surprising, by the way, is a very good word for a patent examiner, that clearly demonstrate the opposite. By the way, I should point out that we also showed for the first time that you could use drug delivery for things like RNA, DNA, as well as proteins and so forth. At any rate, we showed this to the patent examiner, and he did allow the patent. And the next slide shows an example, shows that. And it was a very, very broad patent. And with that, I got involved, and the patent was very key to getting the investments and getting companies interested. So with that, we taught different companies how to do it. We licensed it to companies. I got involved in starting companies. And the next slide just shows a number of the products that have all been FDA approved based on this or related things. And it's a huge number. I mean, there are peptides like leuprolide acetate that people wanted to use for prostate cancer and endometriosis, but they couldn't because you couldn't swallow it, for example, or put it in a patch. And if you injected it, it was destroyed right away. So they put it in these little polymer systems. This is Takeda and others, and then who we had an arrangement with. And then they would inject it and it could release the drug. Initially, they made it last for a month. Now it's 6 months. And there are many, many others. There's treatments for schizophrenia, for different eye diseases, drug-eluting stents. These are some of the major ways of treating opioid addiction, treating pain, diabetes, and so forth. And tens, if not hundreds of millions of people use these. We also went further and even made nanoparticles. And one of the things was to make sure they didn't get destroyed or eaten right away. So here on the next slide, we made little nanoparticles. We published this in Science. We coated it with polyethylene glycol, and you could put the drugs in. And you could either use lipids, or you could use polymers to make these. And you could even, as shown on the next slide, put targeting molecules in to make it go to a certain place. And I thought I would just show you a video. Actually, Nova TV, Nova, which is on the public broadcasting station in the United States, they made a video on some of what we did. I'll just show you the video on the next slide. They explain it better than me. Can the sound be turned on? So this is, like I say, a video, and what you see is, of course, it doesn't blow the cell up that way, but you can use these techniques that we developed to deliver small molecules and even siRNA. In fact, Alnylam, a company that I was on the scientific advisory board from the beginning, used the nanoparticles to deliver siRNA, and that's been used now, and it's been approved to treat a disease, ATTR amyloidosis. That's a very bad disease, by knocking down a gene. And then, of course, Moderna, that's a company I helped start, is using it for delivering messenger RNA. Moderna's in 13 clinical trials, and of course, one that people may have heard about is it's the basis of the COVID vaccine. In fact, some other companies are using that kind of approach too for vaccines. But that's been very exciting, and again, this is again an example of of combining, biology and engineering to create new technologies, and without the combination of both the biology and the technology, I mean, we wouldn't be where we are in terms of the COVID vaccine. I thought I'd give a couple of other examples of things we did, again, to illustrate this point about combining science and technology to make important innovations. So along with a couple of my postdocs, Ram Sasaskaran and Ganesh Venakatraman, we came up with ways to isolate different glycoforms of polysaccharides. The next slide sort of illustrates that. And what we did is, like, heparin's a good example, but really any glycoprotein, this will be true of. The proteins, of course, if you look at a protein, it's basically monodispersed. Every molecule is the same. That's not true for polysaccharides. Heparin, for example, is really a combination of different molecules. So basically, we did this based on sequencing approaches. We came up with the first sequencing approaches to create polysaccharides. This is what two of our postdocs did, and uses a combination of mass spectroscopy, mathematical modeling, and we have molecular scissors, enzymes, heparinases, that we actually had isolated. I did that myself, and that can chop these different molecules up. You analyze those by mass spec, and you put them back together with the mathematical modeling. And we started a company, this is shown on the next slide, to make different products, heparins, other complex polymers, and new glycoproteins. And Momenta, which is the company, there's a profile in the next slide. Like I say, we started with two of our students. We went public. We got major investments from 4 different companies. But then what was exciting is that we got the first Lovenox. This was actually, that should have been 2010, not 2018, first approval by the FDA of any company. And we also got Copaxone approved by the FDA. And then Johnson & Johnson acquired Momenta actually just a couple of weeks ago, not only for these drugs, but we also, using this kind of knowledge of polysaccharide sequencing, we were able to create new autoimmune treatments now in clinical trials for diseases like myasthenia gravis. So Johnson & Johnson acquired it for $6.5 billion. But there's one other thing I wanted to go over, which is the good that can come out of all of this. And that is that about 12 years ago, there was this terrible heparin crisis. The next slide illustrates some of that. I'm sorry, before that, I just wanted to show the product. There was this terrible heparin crisis. The next slide will also show Momenta did well after the approval. But at any rate, on the heparin crisis, what happened was, this is shown in the next slide, that a lot of people died because there was contaminated heparin coming from China. And I had been chair of the FDA Science Board, their highest advisory board, for a number of years. And Ram, my student, had done a lot of work with them. So they asked if could we help. And what we did is we used the sequencing approaches at the company, and at that we developed at MIT, and we published 3 articles. The next slide shows those. The bottom one was isolating the contaminant that was killing people. It turned out to be an oversulfated chondroitin sulfate. But we also published 2 additional papers with the United States Food and Drug Administration and the Center for Disease Control, showing that when you, that this was the cause. These were all in the New England Journal of Medicine. And after we did this, there were no more deaths. So that was actually very exciting and very important in terms of saving a lot of lives. So the next thing I wanted to go over, also in combining life sciences and technological innovation, is an idea I had with Jay Vacanti that really leads to creating new tissues and organs. And this was very controversial again when we first came up with it. But let me just show you a picture on the next slide of one of the ideas that we had, and so that we could make organoids and tissues and things like that. But our idea is that we could take any cell type. In fact, we did this. We proposed this originally in the 80s because nobody talked about stem cells then. But now, of course, you could use these principles with stem cells or iPS cells. But the idea is you could take the cells, you could dissociate them, you could put them on a polymer template that will ultimately dissolve, grow it in a bioreactor, and then literally make any new tissue or organ. One of the ones that we made that's most advanced, in fact, that's approved and now used in a number of situations, is to make new skin. And here, I'm going to show you a baby who's very badly burned. Next slide. You can see the burn. And then you can take the skin and you can even cryopreserve it. It's shown in the next slide. And then if you put it on the child, next slide. I'm sorry, this is at time 0, so you can see still burned. But now let's come back 3 weeks later. Next slide. And he's doing better. And let's come back on the next slide 6 months later. And he's totally healed almost. And these have now been approved by the regulatory authorities for patients who are burned and have diabetic skin ulcers. And in fact, therapeutically, myself and my students, and now lots of other people who are working in this field, have made all kinds of new organoids in vitro and in vivo. There's ones in advanced phase 3 trials for treating, for making new blood vessels. There are clinical trials for making new spinal cords. There's clinical trials for encapsulating cells. So this has been a very exciting example of combining life sciences and technological innovation. I should have also mentioned we're in clinical trials for hearing loss with a slightly different strategy. But I thought I'd go over one other example, which is that you can make these organoids on a chip. And I'll just pick an example where some of our students did, Milika Ratzik and Gordana Vanyak, and we actually started a company, I'm a director of it, called Tara Biosystems. And basically here we're taking cardiomyocytes and you can put them on the scaffold. And the next slide shows that you actually get these structures with the sarcomers and intercalated discs, which is exactly what the heart structure looks like. And then I'm going to show you a video where we took 6 million cells, 6 million cardiomyocytes, but we're going to put them on the scaffold and hopefully it'll beat like a heart. Let's go to the next video. And sometimes I make the joke that people think we're shaking it, but we're not. It's all beating together, 6 million cells, just like that. And you see this. And if I go to the next slide, where you can even take an EKG of it. If we go to the next slide. So you can actually take an EKG, and people are now using this for drug testing. Other people are making livers on a chip, all kinds of things on a chip based on these kinds of principles. So I thought I'd end by by sort of summarizing on the two questions that I mentioned in the beginning. If we go to the next slide, why is it important to strengthen the bridge between life sciences and technological innovation? Well, sometimes you don't, I think all the examples I gave hopefully illustrate that, but I wanted to expand on one, which is this COVID crisis. That's a tremendous example when you look at what's happening, combining nanotechnology, which we've done, with messenger RNA to really, for the first time, create a whole new class of vaccines that Moderna and other companies are now doing. But it wouldn't have happened without the understanding of life sciences, but also without the technology. And there are many, many aspects to that technology, to that nanotechnology. And I hope it'll lead to a vaccine that'll make the world a lot better place. The second thing, and certainly I'll get to this more on the final slide, is the economy. You know, what's happened in the United States is there's a tremendous biotech industry. Moderna and Alnylam, the two companies that I mentioned, working with nanotechnology to deliver siRNA and messenger RNA are two examples. Momenta is another, but there's hundreds of examples. And I think that it's just changed the world because of all the positive drugs and diagnostics and medical devices and everything else that has come out of it. And there's much more to be done. So finally, I just would go to the last slide, which is what can Scandinavia potentially learn from the US ecosystem? And again, I've gotten this more from people I know in Stockholm and Lund and other places, but some of the things that I think go on at the US universities, which I think are very helpful, are, you know, really getting into entrepreneurship and technology transfer. Like they have whole departments in a number of the US universities, MIT and Stanford being two great examples that have led to many companies and a huge effect on the economy, have offices that really understand the business and create great relationships with companies. It also brings in funding, but I think it's really led to, you know, licensing our patents to companies and in sending those companies to really make great products. And another thing, of course, is intellectual property, which of course is done in Sweden as well. But I think that the universities make a big deal out of making sure you get intellectual property, because that will help you go to the last thing, which is getting funding, whether it's from companies or this whole venture capital community that we have in the United States, which I think has happened in part because of laws in the US like the Bayh-Dole Act and the difference in capital gains tax compared to ordinary income taxes, which provides incentive for people to want to invest in that. So those are some things that might be helpful. But anyhow, just in summary, I hope that that what I've gone over illustrates, I think, some of the challenges and some of the things that may be important in trying to combine life sciences with technology and innovation. And like I say, there's an awful lot more to be done. What I've said is probably just the tip of the iceberg. Thank you very, very much for listening to me. It's been a pleasure and an honor.